Boom!
You're still here? It's over.
Yeah, I don't know why it's saying it's over. It's only just started. Yeah, welcome to Let's Get to the Marks. Gonna go through photosynthesis for A-level biology. I think I need to put my hat on, otherwise my bald is going to blind everyone in the... Oh, well, never mind. Okay, what am I going through today? I'm going through photosynthesis. Would help if my head wasn't cut off. Hope everyone's cool. Hope you can hear me on the sound and everything. So, let me go, let me go, let me go. What are we doing? Let's have a little look. What have I got there? I've got a chloroplast. So, why don't I start with a chloroplast? That would make sense, right? Let's go back here. Let's go, let's go. Oh my days. Smash a quick image of a chloroplast down. Give the game away. Probably should review that first of all. So, doing photosynthesis today, guys. Yeah, any questions, just put them in the chat. Anything you want to know about biology, whatever you want to know, I can go through. But glasses, where are the glasses? Okay, I've got like 22 pairs of glasses, but let's lick this on. Okay, nice. I saw the garlic cells in the anaphase, not prophase. Ah, yes. Shout out to Mathilde. Well done. Nice. Yeah, yeah, yeah. You can see all the phases really clearly. That's awesome. Have you guys covered photosynthesis at all? Do you know any of the structures in here? Who can tell me in the chat any of the structures? What we got going on inside the chloroplast? Of course, chloroplast once upon a time used to be, well, their ancestors were eukaryotic cells, just like our ancestors, and for some reason, symbiosis, endosymbiosis, they got inside some of our ancient ancestral cells, and symbiotic relationship, they got shelter, and they provided the ability for those cells to photosynthesize. So, yeah, still around to this day, those there we go. Right, they contain 70S ribosomes. Yeah, that's evidence that they were once related to bacteria, isn't it? They contain those small ribosomes just like bacteria do. Sorry about that. The grabber, the grana, you're right, the grana, they have grana and thylakoid membrane. Nice. Yeah, yeah. Good, good, good. So one of these, just an individual one of these is a thylakoid. So one of these bad boys is a thylakoid. That's a thylakoid. And that is key. The membrane of the thylakoid is where lots of the photosynthesis action takes place. Of course, we've also got, we call a stack of them, like a whole stack. If you were to like check out that whole lump there, we'll call that a grana. So often, grana and thylakoid are on the same marking point in your exams. Thylakoid means one flattened fluid-filled disc, and a granum is a whole bunch of them. Do you know what we call... I'm going to give up on the typing, I think. Do you know what we call sometimes these stacks of thylakoids are joined together by a membrane in between two stacks? Have you heard of the membrane that joins them? Oh, I think Mathilde's mentioned it. Lamellae. So we call those intergranal lamellae, and sometimes there's a membrane. You can just about make out one here going. Let me just put a little line there so you can see it purple there. I'm just drawing a little bit of purple. You can see that is something combining called the intergranal lamellae. It's where the membrane of the thylakoid is extended out in a long extension and joins onto the membrane of another thylakoid in an opposite granum. Yeah. Yes, you would find starch granules, these things. And of course, during photosynthesis, plants make glucose. And the glucose isn't immediately used for respiration. Some of that glucose is turned into starch granules. And so you would expect in the fluid or the gel-like substance found inside the chloroplast, you would expect to find starch granules, which are glucose molecules joined together. You will find some lipid droplets. Why is that? Because if you take a glucose and you join it to fatty acids, you can make a triglyceride. Glycerol, 3 fatty acids, is not far off. If you take some glycerate 3 phosphate, which are some of the things that we use to construct glucose, plants use to construct glucose, you can take the glycerate 3 phosphate and instead make lipids. Cool. So you would find lipid droplets. There are some amino acids you can make from glycerate 3 phosphate as well. However, what was I going to ask? One keyword. Oh yes, Mathilde did already put it up. The stroma. So the gel fluid substance, the stroma, is there as well. So nice. So that's your chloroplast. Brilliant. And you got quite a few of these in plant cells and green plants. Don't be mistaken, you don't get chloroplasts in photosynthetic single-celled organisms. So you might find chlorophyll, the pigment, one of the primary pigments for photosynthesis, inside algae or inside cyanobacteria, but you won't find chloroplasts. A bacteria is about the same size as a chloroplast. You couldn't fit a chloroplast in a bacteria, so they might contain chlorophyll, but they won't have a chloroplast inside them. Okay, let's move on. So what we're going to look at, photosynthesis, super important reaction. Obviously, animals, without photosynthesis, we wouldn't be able to eat plants and other animals and get those stores of glucose, protein, and lipids that the plants have made. So plants make protein, lipids, and glucose via photosynthesis, complex organic molecules. We as heterotrophs, they're autotrophs, we as heterotrophs go and eat plants and other animals, and we get all of our resources, protein for building muscle fiber and tissues and structures around the body, and of course we get the glucose for doing aerobic and anaerobic respiration. So we're going to look at photosynthesis. It happens in 2 reactions. Do you know the names of the 2 reactions? Oh, Mathilde is on fire coming out with all the key points. So I'm gonna have to give you a yeah. Well, if I found my boom button, you get a boom. Boom. There we go. Okay, so yes. Oh, check out the camera. I need to move my head down a little, maybe, potentially. Maybe, whatever. I think I've cut my head off. That doesn't matter. Yeah, we've got the light dependent reaction. Don't move the screen around, you idiot. Right. We've got the light dependent reaction and the light independent reaction, and I'll be going through those today. So be going through both of them for you guys. So who we got here? Yep. So big shout out to Mathilde, answering tons of questions. #1 chat person at the moment. So let's go and get on. Right, let's go through the light dependent reaction. So what we got going, we got the light dependent reaction here. What's this all about in A-level biology? So this is part of your photosynthesis. This is going to take place on the thylakoid membrane. So this is going to take place right here, and this is a zoom in on the thylakoid membrane. Just to remind you of what the thylakoid was. The thylakoid is over here, and it is, okay, just mock up my thing, but the thylakoid is right here, and this is where the first reaction takes place. We call it the light dependent reaction. I'm gonna have to change my screen because what's happened is it's bodged along. There we go, lovely jubbly. And going back, this is where the LDR happens, the light, not the light dependent resistor like in GCSE. You don't have to go back to that, but light comes down, a beam of light hits here, and we're going to look at all the reactions that take place in the light dependent reaction. The main things for you to take away from that are what the products are of the light dependent reaction. It's a quite detailed mechanism. I'm going to spin through it and then give you some questions on it, but the main thing to take away is what are the 3 products of the light dependent reaction. And then of course we have something called the light independent reaction, and that takes place in this jelly, this clear area around here where you see the starch granules like that. Okay, in my day, when I did A-level biology way back in the 1990s, we didn't call it the light independent reaction. We called it the dark reaction. It's also called the Calvin cycle, but we called it the dark reaction. Now, it was probably a stupid name because it can happen in the light or dark, but it doesn't require light. That was the main thing to take away from that. But they no longer call it the dark reaction, probably because the name was a bit misleading and made students think it only occurred in the dark. Okay. Hello Galaphus, big shout out to Gala. Yes. So let's get on with the old light dependent reaction. So what's going on? We have 2 systems, Two systems made of a protein and a pigment. Does anybody know what we call the system that is made of a protein and a pigment? There are two of them, and they're found in the membrane of one of those flattened discs called the thylakoid. What are those called? And do you know the order they come in? So these are a key part of the light-dependent reaction, these structures. They're made up of a protein with a pigment bound to it, okay? Yes, you are right. We have photosystem 2, which is the first one, and the second one is photosystem 1. I think of them like PlayStation 2, PlayStation 1, whatever. Boom! Well done, Gala. Oh, we got user CDOP41, and I can't say the rest. That's too long. There in the chat, big shout to you. Yes, we got PS1. Let me just write that on here. Let's scribble on here. So we've got PS2 and PS1. We'll call this one PS2. Anybody know the wavelength of light? And I think it's a bit specific. You probably will never, ever, ever be asked it. But there is a specific wavelength of light that PS2 absorbs, and it is 680 nanometers. So it will absorb light in the 680 nanometers width of light. The narrowest light gets is about 200 nanometers, but this can absorb light rays that are 680 nanometers wide, and PS1 is down for absorbing light waves or rays, whatever you want to say, that are 700 nanometers wide. So two different photosystems. They're made of a protein with a primary pigment attached, and that primary pigment is chlorophyll A, but there are some other pigments. What color would that be? We'll check that out, Gala. I got a little thing there of what color they would look like. But there are also accessory pigments. So the primary pigment in photosynthesis is chlorophyll A. Has a magnesium a magnesium atom or ion at its reaction center, and it's there that the electrons get excited and leave. But there are accessory pigments, Gala, that absorb light in different wavelengths as well. You've got chlorophyll B and beta carotene. Now these pigments, beta carotene and chlorophyll, their electrons do get excited, but you don't go in at A level into the detail of how they pass their energy on to chlorophyll A. Essentially, chlorophyll A is the main pigment, the main thing that contributes electrons to the electron transport chain. The other accessory pigments, which are carotene and chlorophyll B, they will, through a series of something called electron funneling, but you don't need to know that now, or tunneling, funneling, whatever, they pass their energy on to chlorophyll A. So they just enable the photosystem to absorb different wavelengths of light. And I do have, to answer your question, briefly going off, do have like a little chart like that will show you the kind of the color ranges. So look, chlorophyll A, that is really going to be the main pigment in photosystem 1 and 2, because you can see there that's around the 680 to 700 nanometer wavelength. And then you've got, they do contain these other accessory pigments, those photosystems, that are absorbing a little bit of light in other ranges. So it broadens the spectrum of light they can absorb, and it means it makes it more efficient. They can use more of the sunlight than just that narrow little range there. But we'll come on to that a bit later. Let's just smash this bit out here. So back here, what's going down? I'm going to just talk you through this. Of course, you could make a thing of it if you really want to. Right, this is the stroma. This is inside the thylakoid, and this is the stroma inside the chloroplast, and this is inside one of the discs. So along comes light, and it strikes photosystem 2. Now, yes, Mathilde, there is a practical to separate the pigments. We do thin layer chromatography to separate the pigments, and normally with like dissolved in a solvent of like cyclohexane, propanone, and various other things. And it is literally like GCSE, except for you're not using paper and you're not using a beaker. But it's basically very similar to GCSE thin layer chromatography, as in you'll calculate the RF value, then compare it to known pigments, like known data on pigments, a data book, and identify the what pigments are in there. But for your kind of practical in photosynthesis, you have to know how to grind the leaves up. What is it? Sodium hydrogen carbon? I can't remember, but there's various chemicals that they grind it up with in order to get all of the chlorophyll out of the leaves and stuff. And then you have to carry out the thin layer chromatography. We can look at that. Yeah, so first, right, so light ray absorbed by PS2. What happens next is an electron is excited. Now, this is excited from a magnesium atom. You might remember from GCSE that plants need magnesium to prevent chlorosis, which is when the leaves go yellow. Magnesium, important mineral, absorbed in the roots by active transport. Waterlogged soil, going to make a problem for absorbing these minerals. Magnesium is needed inside chlorophyll A. The light excites some of those electrons from the magnesium and it leaves the photosystem positive. It leaves a magnesium ion behind. Those of you that do chemistry, when you lose an electron, you become a positive ion. We call this photoionization. Okay, that's a key word you can get down if you need to. So the electron gets excited and it enters something called the electron transport chain. Now I've just shown it here as three square black boxes. Now there are all sorts of diagrams and different ways to visualize it, but essentially it's a series of proteins, and as this electron is passed along those proteins, it's got excited, it's absorbed the light energy, and it's gone to a higher energy level, meaning that it's moved through the shells of the magnesium atom, it's moved up through orbitals, and now it's falling back down, and it's passed along these different proteins, and as it falls back down, it uses that energy. That energy is used. Anyone know what that energy is used to do? Yes, this is chemiosmosis, Mathilde. This is exactly the same as we talked about a little bit to do with mass transport and loading into the sieve tube with the hydrogen ions. Very similar. It's exactly the same, and it's found in respiration as well. So these hydrogen ions, what I probably should make a little let's make a little sort of I'm going to need some sort of protein channel or whatever for the hydrogen ions to get pumped through. They're going to get pumped through against their concentration gradient. So what that means is there are more hydrogen ions inside the thylakoid than outside, and yet we still want more. We want more. We want more. We want more. We want tons. We want maximum hydrogen ions inside the thylakoid. And so the whole reason for absorbing the light excites the electron falls and releases that energy, which is then used by a protein in the membrane to pump hydrogen ions into the thylakoid. And then what happens? These hydrogen ions go up through, they diffuse by facilitated diffusion through this enzyme called ATP synthase. Now, the kinetic energy of the hydrogen ions bumping in that passageway, like bumping along, provides enough energy that this ATP synthase, it gets some kinetic energy and it spins, and that provides enough energy for it to join ADP. Is my fat head on that? Yeah, probably. ADP to PI, like that, and to produce ATP, adenosine triphosphate. And that is our first product there being made. So there are three things. If you only remember three things from this, if you remember the name of the three products that are made, you're going to be able to answer some of the questions on the light dependent reaction. So it's all about exam technique. Trying to grasp it all in one go can be difficult, but if you learn that, we get this chemiosmosis, which is the using of the energy released from the electrons that are excited there, passing hydrogen ions being moved against their concentration gradient, which is going to use that energy. So we're not having to use ATP here. This is just the energy created by the electron transport chain. So they're getting it kind of for free, well, from the light. And then those hydrogen ions diffuse back through here by facilitated diffusion down a concentration gradient. And that provides the energy to join ADP and PI, inorganic phosphate, to make ATP. So photosynthesis making a bit of ATP. I know what you're thinking. Is it like respiration? No. This ATP is not going to go on to be used for contracting muscles. Plants don't have muscles. It's not going to be used for all of those things. It's going to be used later on in the second stage of photosynthesis. So what we have to learn here is that the light dependent reaction makes three products, but these things play a vital role in other stages. Well, one of them's waste, but the other two play a vital role in the next stage. So ATP's made. Now, okay, look, our hydrogen ion, what happens next to this hydrogen ion? So this hydrogen ion that's come through here and helped bind ADP and PI to make ATP is going to go on to join with this molecule NADP. Does anybody know what type of molecule NADP is? You'll find a similar molecule called NAD in respiration, and this is called NAD and they're NADP, and they're a specific group of molecules. It's a type of enzyme. It's a coenzyme. That's right, user CDOP41. Boom. It's a type of enzyme called a coenzyme, and its job, a coenzyme's job, is to transfer, rather than having an active site like an enzyme. So it's totally different. It's not breaking things down like an enzyme. It's not substrate, lock and key and juice fit. It doesn't do that. A coenzyme is a transfer molecule. So it is going to carry this hydrogen ion. They're going to join together, and they're going to make NADPH. You can call it NADP reduced, and that's what they tend to call it at A level, which is a bit annoying. I always find it easier, or they call it reduced NADP. I find it easier to call it NADPH because it's just like you've added the H on. But obviously this helps you understand that when you gain hydrogen, you're reduced. When you lose hydrogen, you're oxidized. Okay, now why is it not NADP plus? What's happened to this plus? That's right, coenzyme, you're right. What happens to this plus on the hydrogen ion? Where's that gone? We need something to neutralize that plus. Otherwise this would be NADP reduced plus, or NADPH plus. So what's happened to that positive charge on that hydrogen ion? And it comes back to this little electron that we last left over here, this electron that is now down by PS1. Another ray of light. Thank you. Well done, user CDOP41. Another ray of light excites the electron that's just about lost all its energy. It's finally reached PS1, and it's like, oh, I lost all that energy from the first light beam. Can I go back to PS2 where I belong? No, you can't. What's going to happen is another ray of light will hit PS1 where this electron is, and further excite this electron, excite it once again, and this electron is going to be excited up, look, it's got a negative charge, joins with the hydrogen to make a hydrogen atom, joins with the NADP, and we have NADPH, or you can call it NADP reduced or reduced NADP. And that is the second vital product of the light dependent reaction. So if you had to remember anything, all of this is complex, and I've used lots of terms, chemiosmosis, etc. I'm not going to type it all out because it's too long, but essentially NADP reduced and ATP are your two products of the light dependent reaction. Without those, the dark reaction, or the Calvin cycle, light independent reaction, will not work. Okay, and we're going to look at that because you have to be able to apply your knowledge to say why the light independent reaction won't work. It's independent of light, but if I take light away, why won't it work? So we might think the light independent reaction doesn't need any light. Well, if you take light away from a plant, then you can't get these, and then that reaction can't happen. So light independent, yeah, it kind of does depend on light, even though we call it light independent. Okay, indirectly it depends on light. It depends on the products of the light dependent reaction. Now, we have a problem. These electrons that are passing from PS2 to PS1, well, there's not an unlimited amount of magnesium atoms in this photosystem 2. And if they keep losing their electrons, we're going to run out of electrons. And then the plant is not going to be able to make any more NADP or ATP. So how can we restock electrons here if they keep going here and ending up joining NADP reduced and going off into the stroma away from the thylakoid? How can we restock the electrons in PS2? Anyone know? H2O, nice one, Gala, nice one, user. Let's give you a boom for that. Well done. We're going to need the old boom. So we need H2O. That's right. We've got to get some water going. And so, yeah, where's our water? Well, our water, of course, has come in through the roots via osmosis. And, I don't know. Let's put our water here. That's a fat water. Maybe that pen size is a bit fat. Let me just... Okay, and the water is going to be split. What's it called when water is split? Photolysis, nice. Electrons, water. Thank you very much. Yeah, so we're going to get 2 hydrogen ions. Now, you might find this a bit weird, but because oxygen is a diatomic molecule, if you're a chemist, you'll understand, we have to write half an O2. Oxygen, it's impossible for an oxygen atom to be on its own. So when I write half O2, the misconception is that that's a single oxygen atom. It's not, because this is talking about molar ratios. So that might be, I write 2H plus, that means I might have 2 million H plus. And then if I say I've got half O2, that means I've got like, whatever, 500,000 oxygen, still joint OO, still joint as O2, but I've got half a mole of oxygen. So 2 moles of that, half a mole of that. Anywho, longness, 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 just know to write it like that. And then plus 2 electrons. And I don't know why I'm writing the electrons down there. Those electrons are going to go up here and restock our photosystem 2 without water. So now we need to know application questions called AO2 in biology, whether you're doing OCR, EdXL or AQA. Your application questions are AO2, assessment objective 2. So assessment objective 1 is knowing what is photolysis. Photolysis is the splitting of water by light. Did I show light? No, I didn't show light. Let's show some light. All right, so photolysis is the splitting of water by light to form hydrogen ions, oxygen, and electrons. That is AO1, and that will get you a C grade. If you just get all your AO1 right, you probably come out of your biology A level with a C. So that's memory recall, testing your content. AO2 is application. An application will be something along the lines of explain how the light-dependent reaction will be affected if a plant is deprived of water. So now what we want to say is, what can't happen if I don't have electrons being resupplied here? Which part of this is going to be interfered with? And that's where we have to apply our knowledge and go, right, it releases electrons. There'll be no electrons for the electron transport chain, which means that we don't have energy for chemiosmosis, which means there's no ATP being formed, and there's no electrons to reduce NADP to NADP reduced. And of course, if there's no chemiosmosis, there's no water. We've got these hydrogen ions, which are resupplying hydrogen ions here as well. So we'll have no hydrogen ions to reduce NADP. So you can really go into how a lack of water will F a plant up. Okay, and then a different way, how will a lack of light screw a plant up? Well, we can't have photolysis. So then you could say no electrons, no electron transport chain, no hydrogen ions, chemiosmosis, etc. And then like no excitement of electrons, you get the idea. Yeah, no energy for chemiosmosis. So you need to be able to link off of your content onto your application. Right, last thing, the last product of photosynthesis, and we're moving off the light dependent reaction now. Thankfully. So let's finish with the old light dependent reaction. Is oxygen, and oxygen is a waste gas. All right, so oxygen will be released out of the stomata. It's not really needed later on here. Now, the plant is also respiring, and if you've covered aerobic respiration in oxidative phosphorylation, not all of the oxygen released by the plant by photosynthesis will come out the stomata. Of course, some of it may well be used as the final electron acceptor in oxidative phosphorylation in aerobic respiration. Anyhow, this here, what I've scribbled on here, is known as non-cyclic photophosphorylation. Now that's a disgusting term, and does anybody know what that means? Non-cyclic photophosphorylation. What does that mean? Why non-cyclic? Oh my days. It's got to be one of the worst terms in biology. I don't even know if I've spelt that right. So what does non-cyclic photophosphorylation mean? Well, I'll go through and explain it. The electron, think of our little electron we started with all the way back over here. We started with this little electron here, okay? And this electron in photosystem 2 got excited. It went on a little journey. It went to PS1, then it got re-excited. Woo! joined NADP, became NADPH. That electron is now in here, and that is off to the stroma to get involved in the Calvin cycle, or the light independent reaction. This is not a cycle. That electron has gone. Okay, it's not come back. So that's the non-cyclic bit. The photophosphorylation, well, light was involved. Light was used to excite that electron, and phosphorylation is the ADP plus PI. The ADP, adenosine diphosphate, has been phosphorylated by the inorganic phosphate to become ATP. Anytime we use the word phosphorylation, it just means adding inorganic phosphate to something. So it doesn't necessarily have to be ADP plus PI, but anytime you add a phosphate to something, that's phosphorylation. So this is non-cyclical, photo meaning light, phosphorylation meaning ATP is made. Coolio. Yes, it isn't a cycle. Right. Very, very quickly, and I'm going to show you the cyclic photophosphorylation, and it will take about 30 seconds, so don't cry if you've been listening and you're like, oh my gosh, I can't take no more. So this is going to be cyclic photophosphorylation, and that's underlined in red. Phosphorylation. No, I don't know what I've done there. Anyway, yeah, that looks disgusting. Yeah, so with this one, let me show you what is happening. What happens with this one is a light ray strikes here, and an electron that is in PS2. Remember the electron that we were talking about earlier back here? So there's my lovely electron. If I go back here, this electron came from PS1. That electron came from PS2. That electron came from over here. It doesn't belong to PS1. Let's come back over here now. Right. Now, a separate light ray, maybe, potentially, is exciting an electron that is from PS1. And this only happens with PS1. This does not happen with PS2. And this electron basically does this, hits an electron transport chain, falls down, and then goes back there. Another light ray excites it, and it goes back. It just goes round in a circle like that from PS1 to the ETC. ETC in capital stands for electron transport chain, a series of proteins embedded in the thylakoid membrane. Okay, so this electron is just being passed along these proteins and back here, then excited again, then passed along back here. So what's the point of that? Well, it means that the plant without water, the plant can still make some ATP, right? This doesn't need to be resupplied with electrons from photolysis. It's providing a little bit of energy to move hydrogen ions over here, and the hydrogen by chemiosmosis, and the hydrogen ion can come up here and do this, right? Now, it provides the energy for ADP to join with PI and make ATP. But it does not reduce NADP. Why? The electron would need to come up here, and it doesn't. So just having hydrogen ions, you cannot reduce NADP just with a hydrogen ion. You need an electron. So what we say is that cyclic photophosphorylation can only produce ATP because the electron is cycled back to the electron transport chain and does not go on to reduce the NADP. So this is an example of how the plant, I guess, with water, if it can't get water or something, it can still make some ATP without photolysis happening. A small amount of ATP, but it can't actually make any reduced NADP. Okay. Would it rather choose, Gala said, would it rather choose oxygen from the environment or the byproduct? Yeah, I guess whatever's closest. I'm not sure the percentages. That's something to look up. Does it use oxygen? I guess it's a mix. You've got oxygen diffusing from high to low in through the stomata at certain times of the day. Depends on the rate of photosynthesis and the rate of respiration at different times of the day and stuff as to where it will have access to, whether it will need oxygen to come in. I guess when it's respiring a lot and at night there's not a lot of oxygen, so the respiration will still be happening at night, I guess, and there's not a lot of photosynthesis, so it'll be using oxygen from the environment. But I guess during the hot part of the day, it's probably producing tons of oxygen from photosynthesis. That's probably getting used during the day for respiration, that oxygen. But at night, it's not going to be producing much oxygen as there's no photosynthesis, so it's going to use oxygen from the atmosphere, I imagine. So it's kind of a combination of both, probably at all times, but certainly time of day will affect it. Right. Okay. Let's go on to the light independent reaction, and then we're done photosynthesis. We can look at some questiones. Okay, so hopefully everyone gets that. This is cyclical. Once again, for you, if you didn't understand it, light ray hits photosystem 1, excites an electron up to the electron transport chain. That electron goes boom, boom, boom. It falls down the electron transport chain, releasing the energy it had from the light. So it's transferring the light energy to various proteins in the membrane, and those proteins pump by active transport. They're using the energy from the electrons, not from ATP. They move these hydrogen ions against their concentration gradient into the thylakoid, and they move by facilitated diffusion through ATP synthase, and that gets the energy to make ADP plus PI. So phosphorylation happens. Light is involved, so it's photophosphorylation, but it's cyclic, and there is no NADP. This is not made in cyclic photophosphorylation, only ATP. You don't get any oxygen made because there's no photolysis. Right, let's move on. Yeah, I think the light independent reaction is easier to learn than the light dependent reaction. But actually, if I'm honest, when you go through the exam questions, I haven't seen an exam question that asks you to like bust that out. Okay, so there's no like, this is good for you to understand. And the way I learned this is to be able to just do this off by heart. So you can learn this while watching the TV. Do you see what I mean? You can learn this while just watching the TV. You can just sketch that out on a bit of paper from scratch. Have your revision guide there to look at if you need it. But just get a bit of paper. Say, I'm going to sketch out the light dependent reaction. And if you can draw it all out, then you pretty much understand it, right? If you can draw that out, that diagram, you'll understand it. So that's my way of learning it. But most of the questions don't require you to know all that. You just need to be able to apply the knowledge. Right, light independent reaction. What's going on here? So I'm not going to do all of the terms because that would just take forever. But carbon dioxide comes in by diffusion through the stomata into the leaf. And then it's going to combine with the most common enzyme in the entire living biological world. Does anyone know the enzyme carbon dioxide joins with? It's the most common enzyme in life. Sorry, just having some Vimto. Bit watery. Okay, yes, it's Rubisco. This enzyme, Rubisco, and it's got a kind of rhythmical name, Rubisco. It's quite a cool name, that one, isn't it? Rubisco is found in like anything that does photosynthesis. Rubisco. So, yeah. And we've got a term for this. This carbon dioxide is being fixed. So some people call this carbon fixation. It doesn't mean you're obsessed with carbon, like I love carbon dioxide. No, carbon fixation is taking carbon dioxide from the atmosphere and fixing it into organic molecules. So we call this carbon fixation, or carboxylation, some people call it. And it's not the only substrate in Rubisco. Another molecule reacts with the carbon dioxide and also is bound by the Rubisco. The Rubisco takes the carbon dioxide and joins it with another molecule. What molecule does the Rubisco join the carbon dioxide with? Hopefully you know this. It's called RUBP, and it's known as ribulose biphosphate. Yes, RUBP. I am indeed. I don't know what it means, but yeah, RUBP. And you have to know, important, you need to know how many carbon atoms are in RUBP. And it's a 5-carbon molecule. And essentially, what does Rubisco turn carbon dioxide? And carbon dioxide is obviously a 1-carbon molecule. So we've now got 6 carbons. This Rubisco catalyzes the reaction, basically joins them together, and it makes a very unstable 6-carbon molecule that you don't need to know about for A level that breaks down and forms 2GP. Now, GP, general practitioner, is what you call your doctor when you have to make an appointment. But this is a slightly harder. This is called glycerate 3-phosphate. I'm not going to write it down because that'll take me all evening. So it's glycerate 3-phosphate, and these bad boys are 3 carbon as well. So each one of them, total of 6 carbons that we made over here. Now comes the bit from the light dependent reaction. So this is going to be, all of this we're memorizing here is our AO1, Assessment Objective 1. But the hard bit in biology, the reason why biology has much lower grade boundaries than A-level chemistry and physics is because you have to apply your content knowledge, and that's the bit that people find really tricky. So this is going to be converted into TP, triose phosphate. Now, if you are doing certain exam boards, call these different things. So call some of these molecules different things. I'm calling them the same as in the AQA spec. So there we go. Right, 2 TPs. Now what happens here is one carbon atom is broken off and goes on to make a complex organic molecule. Now we normally call that glucose. At GCSE you learn about the 6CO2, 6H2O comes together to make C6H12O6 plus oxygen, right? It makes a glucose molecule and some oxygen. Well, we made oxygen in the light dependent reaction. It's in the light independent reaction that we make the glucose. So this one carbon atom goes off to make organic molecule such as glucose. Okay, so glucose is really the example. There are loads of other molecules they can make. They can make lipids, amino acids. It can make all sorts. Okay, photosynthesis is not just making glucose. It's a bit of a misconception. Although it does first make the glucose, these GPs or TPs could be used to make amino acids or something else. Doesn't specifically have to be glucose. That might be the majority. I've not looked up the percentages of the amounts, but maybe that is the majority of what the plants make. I would think so, because they make loads of starch, don't they? So one carbon. So you have to think to yourself, what's the formula of glucose? Glucose is C6. Oh my good gosh. Glucose is C6H12. And then O6. So you have to ask yourself, how many times does the Calvin cycle, or the light independent reaction, have to go round to make one glucose? And the answer is 6 times. Might seem pretty inefficient, right? It's got to go round 6 times. And I haven't finished this yet. So, and then of course, what's going to happen is the other 5 carbons are going to be over here, and what we're looking to do here is regenerate RUBP. So only 1 carbon goes round, the rest go back to what they were at the start. So you would say this seems a pretty inefficient process, right? Pretty inefficient. We've gone from RUBP, joint with carbon dioxide, we've turned to GP, we've turned to TP, and only 1 carbon atom goes off to make glucose. The other 5 go back there to continue it round. Now, here's the application bit. If a plant doesn't have light, if you put your little plant in a cupboard and you shut the door because you're like Harry Potter's parents and you lock him under the cupboard, right? You lock your plant under the cupboard. It's not going to get light. Then it's not going to be able to go from GP to TP. Why is that? Why without light can we not go from GP to TP? And these are the kind of questions you will be asked in exams. So to go from GP to TP, this is where we need to bus out our ATP and NADP. Do you remember back here we made NADP reduced and ATP? We need the phosphates and we need the hydrogen ions and the electrons that those molecules are carrying to turn GP into TP. Without that, we can't make TP, we can't make glucose. So going back here, we're going to need 2 ATP, and they are going to become 2 ADP plus PI. So, and we're going to need 2 NADP. I'm not going to write reduce, I'm going to write H. And that's going to go to, now that might not be correct. I can't remember if it's 1 H or 2 H's. Oh my gosh, that is just disgusting. Right, 2 NADP. Okay, so essentially what I'm saying is 2 reduced NADP. They're giving their hydrogens to GP, and ATP is giving its phosphates to GP, and then we're making triose phosphate. So it's being, what's going on here? We've got reduction. These are being oxidized, but we've got reduction and we've got phosphorylation happening. So we've got phosphorylation going on. Now, without the light from the light dependent reaction, if we put our plant in the dark, it can still make GP. It can still get carbon dioxide locked in a cupboard. It just can't turn that GP to TP. So you may be asked questions where you have to apply that knowledge. Okay, so the Calvin cycle or the light independent reaction will still happen, but eventually you'll just have crap loads of GP and no TP. So if you were to look at a plant that was locked in a cupboard, it would have very high concentrations of GP because it can still get carbon dioxide. Eventually all of its RUBP will run out and become GP, and it will have no RUBP left and it will have no TP left, just tons of GP. So you have to be able to apply that stuff. And I believe, but you can correct me if I'm wrong here, I think it's just 1 ATP. ATP. Who's down with ATP? You know me? Right, yeah, right. Plus PI. Okay, so ATP goes to ADP plus PI, and so 3 molecules of ATP. How many molecules of ATP would you need in the light independent reaction to make 1 glucose? That's absolutely right, user. You're right. Yeah, GP needs ATP and NADP reduced to convert into TP. So 3 ATP, and then the TP, the 5 TP that are left, it's not really TP anymore because we've taken a carbon, needs to be regenerated. Yes, you're right, 18. We need 18 ATP to make 1 glucose molecule. Nuts, isn't it? And just think, when an animal eats glucose, if it maximizes the respiration from it, I don't know if it's 32 or 38, but it might get quite a lot of ATP out of it. But just in this stage alone, 18 ATP are needed. And that's because 3 ATP are needed to get 1 carbon for the glucose. You need this cycle to go round 6 times to make 1 glucose. So that would be a total of 12 NADPH and 18 ATP needed to produce that 1 glucose molecule. And that's the light independent reaction for you right there. And what I would do, if you're revising, the best way to revise all this disgusting biology content is literally that diagram there. I might have missed something out, but on the fly, but you can always check it against your revision guide. Take a bit of paper and just say to yourself, I'm going to watch some Netflix, watch some Amazon Prime, whatever your thing is, and I'm going to sit there and chill, and I'm just going to, if I'm just watching something that I don't have to get too deep on, I'm just going to chill and I'm just going to sketch out the light independent reaction, and then I'm going to try and draw out the light dependent reaction. There's loads of parts that you can do like that you can just learn. Once you've learned to just be able to scribble them on a bit of paper when you're not feeling like doing exam questions, then you pretty much know this off by heart by drawing it all out. And you'll be able to see the connections, like, oh, I need hydrogen to make GP into TP and phosphates. And you'll start to understand what phosphorylation means, like adding phosphate. Okay, let's have a little look. Does it take a long time for the cycle to happen in real time? Yeah, I'm not sure how long, what is the rate? That's a good question. What's the rate of glucose production in a plant? Like, as in, how many moles of glucose, how many molecules? I don't know if they've got it down to how many molecules. They probably know how many moles of glucose are produced per hour or something. But yeah, that's something to look up. Right, let me give you guys a few questions. So, couple of questions to do on the lovely photosynthesis. So yeah, that was all the content for photosynthesis that you really need to get down. You can look at, there are some other things to know about photosynthesis. You got to know that having more than one photosystem and having more than one pigment, i.e. having accessory pigments, they're called accessory or secondary pigments, which are chlorophyll B and carotene, they broaden the spectrum or the wavelength of light that can be absorbed, and that's obviously good for the plant. It means it can absorb light, a wider range of light. So like throughout the day, different wavelengths of light can hit the plant, and more of the light that actually hits the plant can be used rather than just bouncing off or not being used. Are we going to see in photosynthesis which chemicals are needed for the light dependent reaction? Does this say which ones are produced or which ones are needed? This is which ones are needed, not produced. So what do we need? We need... So, yeah, needed. So what do you need before you make the ATP? What's needed to make ATP? And what's needed to make reduced NADP? So we're going to need NADP, so it can become NADP reduced. We're going to need ADP, PI, so they can become ATP. And we're going to need water to produce hydrogen ions and electrons. So it's going to be D. Yes. Well done, Oscar. Well done, Matilde. Yes, agreed. We're going to need that last one. Now, of course, when you're doing that and you're like, oh, it said, then think, whenever you're answering questions of physics and maths tutor or Save My Exams, Seneca, whatever exam, always then reverse say, okay, they could ask me what's produced. And then you'd say, right, well, this is what's produced. So it would be oxygen from the water, ATP from the ADP and PI, and reduced NADP. So always ask yourself, you get double by asking yourself extra questions. Describe what happens during photoionization. Ooh, that's a bent line. During photoionization in the light dependent reaction. So what happens in photoionization? What do you reckon? Photoionization, photo means light, ionizing means something. What happens when you become an ion? Yeah, you might remember from chemistry GCSE if you don't do chemistry A level. So electrons are excited to a higher energy level. Absolutely. And we might want to say, and we might want to mention light there as well. You might want to work light in there. Because it says photo, I've got the mark scheme, I'll put it up in a second, but we might want to work light into that answer. I don't believe you need to say electrons from magnesium ions or atoms are excited to a higher energy level by light, but I think you can just say light is used. Light is absorbed, if we want, by a photosystem, and electrons are excited. Electrons are lost. Maybe we need to say a positive ion is formed. We'll have a look at the mark scheme. You can slam it all down. Light energy is used to excite an electron to a higher energy level, leaving behind a positive ion. But essentially an electron is lost, right, to the electron transport chain. A student obtained a solution of pigment, so this is the TLC, from leaves of a plant. The student used paper chromatography to separate the pigments. This is the chromatogram, E, D, C, or A, B, C, D, E. And that's the origin, the starting point. Explain why the student marked the origin using pencil rather than ink. Oh my days, that's like a GCSE one. That one used to come up all the time in GCSE chemistry. Why do we use pencil rather than ink? You know the answer to that. Ink will spread, ink will run, ink will dissolve in the solvent and interfere with the results. It will mask the results, yes, so it doesn't smudge. Ink is soluble. That's ink. Yeah, ink is soluble and may run up the paper and disrupt the results, interfere with the results. Yeah, may mask some of the pigments. Right, what have we got? Describe the method the student used to separate the pigments after the solution of pigments had been applied to the origin. So what they're saying is we've put the pigments on the origin. This is paper, not TLC. This isn't thin layer. So what are we going to dip the chromatography paper in? And what do we need to make sure this... Okay, so we're going to put the paper... I lost the ability to speak. We're going to put the chromatogram... Well, it hasn't formed yet. So we're going to take the chromatography paper. We're going to dip it in the solvent. Make sure the solvent's not above the line. And we're going to wait a set amount of time. We're going to remove it before the pigments reach the end. We don't want the pigments to reach the end. Look, if A goes here... What happens if A gets here? And then I wait another hour and B gets there. That's no good. How do I can't tell them apart now. So we always remove from the solvent our chromatogram or our chromatography paper before the solvent reaches the end. Yeah, I suppose it could be quite accurate, but yeah, yes, you'd increase accuracy, but it's not 100% necessary. But yeah, close to the end, yes. And the reason is that will mean your length, your distance travelled by the pigment is longer, and therefore you've got a smaller percentage error in your results, right? If you're using a longer distance, there's going to be if your ruler has like plus or minus zero point 0.5 error, whatever, then it's going to be a smaller percentage over a longer distance. So yes, probably better to have them go as far as possible. But as I say, you don't want to leave them in there forever, because the whole point is we want a set time, and then we can compare to a data book. Right, let's have a little look at the answers. Oopsie. Yeah, so our answers here, that was right. Chlorophyll absorbs light, electron is excited, good enough. Oh, electrons are lost. The chlorophyll becomes positive, ignore the site molecule from where electrons are lost. So except electrons go to the electron transport chain. So if you just said electrons are excited and they go to the electron transport chain, they're saying that's the same as saying electrons are excited and they're lost. And then electrons are excited by light, or chlorophyll absorbs light. Ink, leaf, pigments would mix. Yeah, this was the one about why should we use pencil? The ink and so the main thing you've got to say here, rather than at GCSE would just say it will smudge, we've got to say what's the problem with it smudging. So we have to say that ink is going to disrupt the pigments, going to interfere with the pigments, like run over them, and that's going to affect our measurements. So you've got to just be a little bit more extra at A level. And then the level of the solvent below the line, remove the solvent before it reaches the top. Okay, cool. Okay, let's zing on. I don't even know if you can read this. This is a bit like busted, so okay. Now this is AO3. So busting out a bit of AO3. No, I don't think that's made it any better. So I'll read this to you. Scientists investigated the effect of temperature on the activity of two enzymes isolated from leaf cells of cotton plants. Rubisco activase, an enzyme that activates rubisco. Figure 1 and 2 show their results. Rubisco activity, as it goes up, or as the temperature increases on our x-axis, the activity of rubisco increases. If we look at rubisco activase on figure 2, as the temperature increases, the activity of rubisco activase increases till about 28-30 degrees, and then it starts to go down after 30 degrees. Now, scientists concluded, they made a conclusion, that heat stress reduces the activity of rubisco in plant leaves by affecting rubisco activase. Use all the information to evaluate their conclusion. So, this is actually an evaluation question. Now, in biology, it tends to be when you evaluate, you look for things that you can rip apart, and you normally want to rip apart stuff about the experiment. You want to say one good thing or two good things and as many bad things as you can, and so okay, very, very subtle. These evaluation questions, like you can revise photosynthesis, you can revise respiration, you can revise your karyotic cells, you can learn your biological molecules off by heart. If you don't know how to tackle evaluation questions, you're guaranteed to lose marks, because they're like a separate topic and you have to apply. The great news is that they're a lot easier. For evaluation questions, you don't have to memorize difficult concepts and electron transport chains, and you don't have to be able to do dihybrid crosses and all of this stuff. You don't have to do any of that. You just have to learn a set of skills that you apply every time you see the word evaluate. So every time you see the word evaluate, you need to apply a set of skills. We need to think, what can we rip this scientific experiment apart. We want to tear it apart. So what we're going to do, we're going to highlight the fact here that you're going to go through the stem of the question and you will find at least 1 or 2 marks in the stem. Look, they isolated rubisco from cotton plants. Okay, they used cotton plants in the experiment. What does the conclusion say? The conclusion says plant leaves, not cotton plants. So these silly scientists, and they do this all the time in AQA and in other examples when you evaluate, someone makes a confident statement applying experimental data to the whole population of plants. Well, we don't know if oak trees behave the same as cotton plants. So they only did it. So one of our bad points is they only did this on cotton plants. They didn't do it on other species of plants, so that would be making the statement invalid. What else? For the rubisco activity, the increments of temperature are wider, which decreases the precision. Yeah, so we can't, yeah, it's harder to note where the optimum temperature is there. Now they isolated the activity of these enzymes from the leaf cells. Another issue I might potentially have is they isolated the enzymes from the leaf cells. They took the enzymes out of the cell. Do we know if the enzymes behave like this when they're in plant leaves? They're in the cell. So they haven't experimented on them in the cell. They've taken them out the cell. For all we know, rubisco doesn't denature when it's inside a cell. It's more fragile outside the cell or whatever. So there's another negative point to make about that. What else do we know? We can see that rubisco is going up there, and then what did they say here? An enzyme that activates rubisco. Scientists conclude that the heat stress reduces the activity of rubisco in plant leaves by affecting rubisco activase. Let's have a little look at the mark scheme. There should be some positive things they can say here. We could say, okay, on the positive side, right? Look, what's positive? Yes, rubisco seems to denature at 30 degrees, above 30 degrees. It looks like rubisco denatures, and that would mean, the rubisco activase denatures, and that means it can't activate the rubisco. So that looks like good evidence that if rubisco denatures at 30, that the reason for the heat stress is this. But what we could say is, however, rubisco is still operating really well at 50, and we don't know. We need experiments where they're together. Like, did they do this experiment where they're together? Because if they were together, then this is having no effect on that, because that is still increasing. What we can say is rubisco activity is still increasing at 50 degrees. So they've only gone up to 50. We don't know what happens after 50. Anywho, these are the kind of things. Let's have a look at the evaluation. So let me put the answers up for you, man, there. Ooh-wee. Oh, go away, chloroplast. You're being replaced by the mark scheme. Okay, so this is kind of rubisco activity increases with temperature. Rubisco's optimum temperature is above rubisco activase. Rubisco activity decreases at high temperatures, or it's denatured above 25. Rubisco activase optimum range is 25 to 30. So that would suggest that heat stress would reduce the activity. Activase does not affect rubisco, but also this suggests what I was saying there is this suggests that maybe it doesn't affect it. This is still going up at 50 even after this is denatured or this is decreased in activity. The results are only for cotton, might be different for other plants. The results are for isolated enzymes, not for enzymes inside cells. And then this is a very typical, this last point is very typical, and they want, whenever you do an evaluation, you can throw this in. Now, half the time it won't be accepted because it depends on the stem of the question, but you can say there's no stats test. Now, we know there's no stats test. Sometimes if you see error bars or you see standard deviation bars, you know, where they put like bars on a graph, then that counts as a stat test. Or they may say something like there was a correlate. They may mention that a stats test is done, and then saying no stats test will not get you a mark. But generally, that's a good one to have up your sleeve. It's like the old repeat 3 times and take an average when you used to write about in GCSE about experiments. So it's like that. No statistical test has been done to show if there's a correlation between these two. Cool. Let's just do one more question before we finish up. Let's do a couple more questions. Oh no, I don't want to do this question. This question's just dirty. Right. I haven't actually explained DC pip, so I don't think I'm going to do that tonight. DC pip. Yeah. So DC pip. I haven't explained this tonight, and I'm getting tired now, so I probably won't bother. But DC pip is an indicator. It's blue in color, and when it accepts, basically, when it gets reduced, when it takes electrons, when it receives electrons, there's a color change. Why this is a nice way of evaluating the rate of photosynthesis is it acts as an electron acceptor. It acts like NADP. So do you remember when we did our, where's my light dependent reaction? When we did this, the NADP picks up this electron, and it gets reduced to NADP reduced. DC pip plays that role of the coenzyme. It basically takes the electron and decolorizes. So what you can do by that is you can set up a colorimeter, or you can, draw an X on a bit of card or whatever and see when the X becomes visible or whatever. You can basically measure the rate of reaction. You can use DC pip to measure the rate of the light dependent reaction, essentially, from the because it will accept electrons and change color. So that's why they use this thing called DC pip, and they can use that with various pigments and stuff. So there's a question here about it, but a student isolated chloroplast from spinach leaves into a solution to form chloroplast suspension. Yeah, you need to know the color change. You do. Yeah, so this says, you probably won't be able to read this on the screen. I don't know why I put this question up. This is an old PowerPoint I had. A student isolated chloroplast from spinach leaves into a solution to form a chloroplast suspension. He used the chloroplast suspension and DC pip solution to investigate the light dependent reaction of photosynthesis. DC pip solution is blue when oxidized and colorless when it gets reduced. Remember oil rig or Leo grrr. Right, Leo grrr is loss of electrons oxidized, gain of electrons reduced, grrr, or you can do oil rig. Oxidation is loss of electrons and reduction is gain, as your mnemonic to kind of remember it. So yes, it's going to go colorless when it picks up the electrons. The students set up 3 test tubes. Test tube 1 had chloroplasts and 9 centimeters of DC pip, and it was left in the light, so I'm going to expect that to go colorless as it picks up the electrons. Tube 2 had chloroplasts, 9 centimeters of DC pip, and it was left in the dark. Basically, can the light dependent reaction happen and the electrons get passed along in the dark? And then tube 3, 1 centimeter cubed of chloroplast suspension, 9 centimeters of DC pip, and that was also in the light. What is the diff? Oh, tube 1 had no chloroplasts. Tube 2 had chloroplasts and in the dark, and tube 3 had chloroplasts and in the light. Gotcha. The student recorded the color of DC pip in each tube at the start and after the tubes have been left at 20 degrees for 30 minutes. These are the results. Tube 1 was blue at the start, blue after 30 minutes. Of course it was blue at the start and blue after 30 minutes. It's the control. We are proving here with tube 1, they could ask you this. Oh, they do ask you it up there, so I probably shouldn't waffle on about it if you wanted to have a go at it. But tube 1 is going to be our control. We're trying to prove that DC pip doesn't go just color. Yeah, I like to go colorless after 5 minutes. So we're just proving that DC pip doesn't change color unless it gets reduced by electrons. So there we go. Number 2 stayed blue. Why? It's in the darkness, so no electrons are excited from the photosystem. And then tube 3 we're going to expect it to go colorless because the light will excite electrons from photosystem 2 and they will get passed along and they will end up in DC pip. Okay, the solution that the student used to produce the chloroplast suspension had the same water potential as the chloroplast. Explain why it's important. Now, if you did the centrifugation topic, ultracentrifugation, homogenation, and all of that when we want to examine organelles, why does the solution and the chloroplast have to have the same water potential? Do you remember your AS year 12 topic about, you use the blender and all of that? Good, Oscar. So the cell, we don't want the chloroplast to burst. We want no osmosis to take place. If we lower the water potential of the chloroplast, water will flood in and it could swell up and burst. So we use an isotonic solution for these things. Now, yes, prevent lysis. Good. Explain why the students set up cell tube 1. I kind of gave that away. That was known as a control. So if you just say control, you'll get a mark. And I guess... No, the chloroplast doesn't have a cell wall, remember. And we've taken the chloroplast out. So the chloroplast has been isolated for this experiment. So there's no cell wall. The chloroplast has just got a double membrane. Doesn't have a cell wall. Yeah. And then I guess tube 1 is the control. And what we would need to say there to prove that DC pip does not go colorless by itself or with light. Because what we're really trying to prove is it's not light that makes DC pip go colorless. See, tube 1, no chloroplast, but light. Maybe DC pip goes colorless because of light, the light energy. But we're trying to prove, no, it's not the light energy. It's something in the chloroplast with light that makes DC pip go colorless. And that's how we can prove that light causes electrons to move and reduce DC pip. Anywho, so this is the answer to the first bit right here. Osmosis does not occur. We don't want it to shrink. And then the next questione, what is the control? Oh, what happened there? Yeah, so to show that light does not affect DC pip, to show that chloroplasts are needed. I thought they might give you a mark for just saying it is a control. Never mind. Explain the results in tube 3. So why did tube 3 go colorless? In case you can't read it, tube 3 has chloroplasts, DC pip, and light. So what do we want to say has happened? Why did it go from blue to colorless? What did it get, and where did it get it from? As the DC pip was reduced. Nice answer, Mathilde. That'll definitely get you a mark. By electrons. Yeah, good. Light. Yeah, the light moved the electrons down the transport chain and reduced the DC pip. That sounds very good. Let's have a little look. They sound like very good answers. Reduction of DC pip by electrons. Chlorophyll, light from chlorophyll. Yeah, so the electrons came from chlorophyll or the light dependent reaction, and yeah. Okay. Oh my God, I can't even read these last two questions. They're so small, let alone on your screen, on my screen. The student evaluated the effectiveness of different chemicals as weed killers by assessing their ability to prevent decolorization of DC pip in chloroplast suspensions. He added different concentrations of each chemical to illuminated chloroplast suspensions containing DC pip. He then determined that IC50, I don't even know what that is, for each chemical. The IC50, I don't even know if that's what it is. Oh, the IC50 is the concentration of chemical which inhibits the decolorization by 50%. Explain the advantage of the student using IC50 in this investigation. So, yeah, that was a big fat stem of a question. He added different concentrations of each chemical to illuminated chloroplast suspensions containing DC pip. He then determined the IC50 for each chemical. That's the concentration of when the chemical inhibits the decolorization of DC pip by 50%. Yeah, we are smashing today, but I need to smash on. So, apparently the IC50, it just creates a standard so you can compare to other weed killers. So what they're essentially doing here, their weed killer, which you spray on plants to kill the weeds, essentially stops the plant from forming NAD reduced, and therefore it can't go on and make glucose and all of that in the light independent reaction. Explain how chemicals which inhibit the, oh yeah, could slow the growth of weeds. I've just kind of said that. So yeah, basically it's not going to, if it's not decolorizing DCPIP, if it's not turning DCPIP from blue to colorless, that means the electrons are not getting to NADH or NADP, sorry, and becoming NADPH or NADP reduced, which means the coenzyme is not bringing electrons to GP and turning GP into TP. So you're not going to get any TP, you're not going to get any glucose formed. And so, yeah, less glucose. So what they're saying here is less or no ATP, less or no NADP reduced. So no GP turned into TP. Yes, that's right, Mathilde. It's only got a certain amount of electrons it can gain. Absolutely. So I can't remember actually. Where's my... Oh. Now, I can't remember if it goes to NADPH2. Let me have a little look. Google is your friend. I know it just goes to... yeah, it goes to NADPH2, so it absorbs 2 electrons. I'm not sure if it could go to NADPH3. I don't think so, otherwise it would. If it could. Let's have a little butcher's. Nah. Yeah, so one of the reasons why in A-level biology, in most of the exam boards, they stopped saying NADP to NADPH is because it can go to NADPH or it can go to NADPH2, where it picks up 2 hydrogen ions and electrons. And so I think they thought that's just confusing the hell out of everybody. Let's just call it reduced NADP. But yeah, it kind of helped people understand that you're reduced when you gain electrons, though, when we used to refer to it as that. Right, I've lost where I am. So yeah, the answer is yes, there is a limit. Yes, so if you increase the amount of NADP, Mathilde, then you will get more NADP reduced. You will get a greater amount of GP converted to TP. But remember, if we go back here, with the GP to TP, there will become a point. It's only if this is the limiting factor. Remember the old limiting factors? It's like making cookies, right? If I could have loads and loads of sugar, but if I don't have any chocolate chips, I'm going to have a limit to the amount of my rate of cookie formation is going to be limited by the ATP. So essentially, I can increase this, but at a certain point, doubling this will have no effect on the rate of reaction until I step up the ATP. So both are needed to go up, but there might be a point where if I have less of one, I increase that one, but then I need to increase that one. So you're going to need to have enough of both, because this GP doesn't just need electrons and hydrogen, it needs phosphates to become TP as well. So yeah, but you're right, NADP, the amount of the supply of NADP and ADP-PI is going to be a factor in limiting the rate of photosynthesis. Oh my gosh, what is this question here? I've got a plant on the screen. Ugh. Okay, so this question says atrazine binds to proteins in the electron transfer chain in chloroplasts of weeds, reducing the transfer of electrons down the chain. Explain how this reduces the rate of photosynthesis in weeds. So what you would want to do here is explain basically if someone interrupts the electron transport chain, how does that reduce the rate of photosynthesis? And you could reword that question for loads of other questions. How does this prevent or slow the rate of growth in the weeds? So if you stop the electron transport chain, what happens? Now this has just said it blocked proteins, but there's other ways to interrupt the electron transport chain, would be to prevent photolysis. So you could have a question saying photolysis is prevented, or you could have a question saying the photoionization energy. Photoionization of photosystem 2 is prevented. How does this reduce the rate of photosynthesis? All of them lead to an interruption in the electron transport chain. So really they want you to what this question is kind of like. This is an AO2 application. So it's not just asking you what does the electron transport chain do. That would be AO1. What does the explain the importance of the electron transport chain? That is AO1. Just give me your content. But this is application. You've got to figure out. So we're going to get less NADP reduced, so less TP made in the light independent reaction. Lovely. What else? Yeah, perfect. That's good as well. But we're also absolutely, and we're also, so we're going to get less glucose made, obviously, as well, if we get less TP. But also, within this question, we might want to explain. Yes, less electrons passed. We might want to say less electrons reduce NADP to NADP reduced. You kind of said that, less NADP reduced. But also, what's the other thing we'll get less of? Look, the electron transport chain is not just for the NADP. It provides the energy for hydrogen ions to be transferred. Yes, Oscar, that's right. Less ATP as well. Why? So we want to say less chemiosmosis. And that's great because chemiosmosis is all of it. So without the electrons, less energy to actively transport or to pump hydrogen ions against the concentration gradient into the thylakoid, which means less ADP joined to PI to form ATP. And if we have less ATP and less reduced NADP, boom, then we get less GP to TP and less glucose. So that's called an AO2 question. You're applying all this knowledge to answer that question. And that's really, so very rare. You can see it'd be very hard. I've searched for questions which are just basically AO1. What does this do? What does Rubisco do? There aren't many of them. Most of them are these application questions. So like if I was revising this, you've got the idea. The key thing to know is this produces NADP reduced, this produces ATP. Yes, all of these other things are important, but that's the key thing. Yes, less organic substrates produced, smaller electrochemical gradient. Yes, yes, yes. That's right. So smaller electrochemical gradient, less ATP produced, less NADP reduced. So less GP glycerate 3 phosphate is turned into triose phosphate, and therefore less complex organic molecules like starch, like lipids, like glucose, like amino acids that are made. And there we go. I think I'm going to finish up. So that was a little one on photosynthesis. I don't know what people want. I was doing polls before, but I'm not sure people want, like I can go through Krebs cycle and things like that in another lesson. We could go through things like the manometer and reactions of respiration. You might want to look at net primary production, gross primary production, biomass. Depends what kind of subjects you lot are on. Of course, I did the nitrogen cycle last week, but of course, you got the phosphate cycle or the phosphorus cycle, where the old guano from the birds, they cycle their poo in the rocks and bird poo. There's all sorts of topics, so you could just leave a comment with which topic you would like to go through. I already did insects and gas exchange and that kind of thing, but then you've got the tropisms, the movement, random movements and stuff like that. We can go for any of that business. Nervous system. So loads of different topics I can go through. Leave a comment in the comments about any topic you'd like me to go through in a future lesson. I will check you lot later because I'm going to shoot off. And I hope you guys have a good evening. Keep working hard, keep practicing, keep using physics and maths tutor to save my exams. All of the resources you've got, making flashcards, mind maps, even when you're chilling out and you're just watching movies on Netflix, Disney Channel, whatever you're watching on. Oh, was my screen all off center? Oh gosh, I hope that wasn't for the whole lesson. Never mind. Yeah, just keep working, and use your time, revise smartly. So draw out the diagrams, and that kind of thing. When you're not in the mood for answering exam questions, just make sure you're making flashcards, mind maps, you're drawing out diagrams and things like that that you can just do. Draw out a eukaryotic cell and label all the organelles, things like that, that you can do that while you're watching, while you're chilling out. All right, nice one, guys. It's time for me to shut up and go, and you guys get on. So take care, and I will see you later. All right, nice one.
You're still here?